Rafael Uribe is from the north of us here at Cal Poly Humboldt State University, and joins us tonight from Arcata. So Noyo Center is really happy to bring you these science talks. And as many of you have heard me talk about before, it's kind of opened up this possibility to get together on a monthly basis with people from all around the world with all sorts of different expertise. Don't hesitate to donate to our organization and continue to show us the support for the work that we do with our scientists and our youth, our interns, our volunteers, and our community here on the Mendocino coast. So thank you. And with that, I introduce to you Rafael Uribe. I said it good before. Uribe. No, you say it. I just botched it. I'm sorry about that.
Yeah.
Uribe. Take it away.
All right. So let me share my screen here. Okay. So today I'm going to be talking about the research that we have been doing with Bull Kelp here at Cal Poly Humboldt. I want to acknowledge my team, especially my graduate students, Erika Kalani, which is here connected, and Marcia, and also my collaborators, Rick Seckman and Karen Gray. And I want to start a little bit more about why I'm doing this project. My background is in aquaculture. I have a lot of experience raising fish, but this project kind of guided me to something new, which is the world of the seaweed. And the first question here is like, what is the #1 species produced in aquaculture worldwide? And the answer, I mean, we are in a seaweed talk, it's gonna be seaweed. So the #1 is gonna be the Japanese kelp, Saccharina japonica, following by Guso, so Eukima. So this is just to set up the stage so you can see the importance of the seaweed in aquaculture. From the top 10 principal species, 3 of them are seaweed. So it's not just aquaculture, it's not just about fish. It's also about other extractive species such as seaweed and oysters. So the seaweed industry has been growing worldwide, since the 90s, 80s. We can see nowadays that we have a production worldwide of 35 million metric tons. And if we compare that with the seaweed that we harvest from the wild, it's not even close, it's like 1.1 million metric tons. So this means that 97% of the seaweed that we use and we consume globally is come from aquaculture. And again, since 1971, the seaweed farms were already producing more than 50% of the old seaweed. Now, most of this seaweed is gonna come from Asia. So just the 4 countries, China being the #1, producing almost 60% of the seaweed worldwide, following by Indonesia, South Korea, and the Philippines. And I want to notice here, Indonesia in 2005, they were producing less than 1 million metric tons. And nowadays, they are the second and producing 9.6 million metric tons. So as you can see, a country that is small was able to really empower that production of seaweed. Now, what about here in the United States? How is the aquaculture industry looking? Well, unfortunately, our aquaculture industry is not looking that great. And it's not because of the lack of funding, because the federal government spent more than 100 million annually but also has been because there has been some critics that are opposed of aquaculture practices. The reality is aquaculture involves a lot of laws. There is more than 1,300 laws that you need to go and get approval if you want to start your own business. And there is an estimation that it's going to take about $1 million and about 5 years just to have your permit in place. So now let's move into the seaweed production in the United States. So the seaweed production in the United States, most of our seaweed that we consume locally is gonna be wild harvest. As you can see, for a long time, we depend on the wild harvest, but lately, you can see that we don't harvest as much seaweed as we used to harvest. And in that little arrow, that's where aquaculture started. And let me zoom in because this is a new industry that is happening here in the United States. So this is the aquaculture industry of seaweed in specific. And as you can see, since 2018 until 2020, this has been grown 5.7 times. Even though we have a nice growth in the industry, but we still rely on imports of seaweed produce. So with more than 95% of the seaweed that we consume coming from other countries. So there is a lot of room to grow seaweed here in the United States. Now, which states are the ones producing most of this seaweed in the United States? Well, it's gonna be Maine. Maine producing 54% of the seaweed in the United States, followed by Alaska. If you follow this table, then you have California, Oregon, and Washington. We don't really produce as much as Maine or Alaska. And, you know, like, why do we want to produce seaweed? Why is the advantage? Well, in your everyday life, you are using something that has seaweed on it. And it could be your toothpaste, it could be your cosmetics. Seaweed is processed into agar, alginate, carrageenans. It's everywhere. And nowadays, there is a new industry developing products that are biodegradable, that are made of seaweed. And we have several companies here in California, for example, Sway, that is producing bioplastic made of seaweed, or straws that you can just dump it into your compost pile, or, you know, glasses that you can also eat after you drink the water. And recently, the new seaweed has been about reducing methane. This is a research that was done by UC Davis, and they found out that Asparagopsis armata is a seaweed that could reduce 50% or more if you feed them to cows. Now, not all the seaweed are going to have that bromide compounds, meaning that not all the seaweed is going to be able to attain that reduction of methane. So that's one of the misconceptions that a lot of people think, that, oh, you just feed them seaweed and it's going to reduce methane, but it's going to be dependent on the species that you are going to be using. So seaweed is going to have a lot of different uses, and depending on the use, it's going to depend on the price. So you can just use as a biofuel and not get that much money, but then you can use even as a cosmetical, or even seaweed. Some of the seaweed, they have anti-cancerous properties so that are used as a pharmaceuticals. So we see overall a demand on the seaweed in the United States. The demand is because people, they want to eat healthy food. And that's why we see a consumption growing of 7% a year. They also want to invest in more sustainable texture additives and overall enhance food security. And here in this picture, I have a company from California. It's called Trophic, where they are incorporating some of the red seaweed into the Impossible burgers or Impossible meat in order to enhance that color of that plant-based meat, so it's become more juicy than just a pale plant-based milk. So why do we want to grow seaweed? First of all, the technology is out there. It's easy, it's cheap. You can produce, or the yields are greater than agriculture. The technology is available, as I say, and the demand is greater than supply. Growing seaweed is going to provide an ecosystem service because you are removing nutrients, you are removing carbon, nitrogen, phosphorus out of the water, and also you are adding oxygen into the water. So at the end, it's providing also an ecosystem service. Let's just talk about here in California. We have more than 700 species of seaweed, but we are the seaweed industry, the aquaculture industry is just in diapers in California. Right now, there is just 6 permits or issued permits from the California Fish and Wildlife to do aquaculture. One of them is pending, and one is an experimental project. So now I'm going to start about talking about our project. We are so fortunate to be located in Humboldt, where we have the Humboldt Bay, and the Harbor District is the institution that manages the bay. For those of you who are not familiar, Humboldt Bay is just across Eureka. You can see here Eureka, and that's Humboldt Bay. Arcata is in the north. That's where I am at the university. So, like I mentioned, we are so fortunate because the Harbor District was able to get all the permits in place and create these pre-permitted leases for people that want to grow oysters or shellfish, clams, but also to grow red seaweed. And they got all these sites available to lease. And that was thanks for several efforts. One of them, Adam, was one of the persons that was in charge in creating these leases. In total, they have about 23.2 acres. But one of the things is it costs a lot of money for them to go into all the institutions and all the laws and get them approved. And because the cost that they spend, also they need to make up the money that they spend. And that's why the lease rate is about $550 per acre per month. So these are some of the sites that the Harbor District has, where our project is going to be located. But before I go into that, I didn't know about this. I didn't know about the Harbor District having these pre-permitted leases. And what happened is in 2018, Moss Landing, especially Luke Gardner, organized this conference where we met and we talked about aquaculture and how we can move forward this mariculture industry in California. During that conference, I had the opportunity to meet Karen Gray. And Karen Gray is the California Reef Manager for Greenwave. And while I was sitting with her in the conference, she asked me like, oh, where are you from? And I say, well, I'm from Humboldt. And she said, do you know that Humboldt is one of the few places that you can grow seaweed in these pre-permitted leases? And I said, no, I didn't know. So she said, why don't we just write a grant and we can start growing seaweed in Humboldt Bay? So the next year, we submitted a proposal to the Agricultural Research Institute to start growing seaweed. In specific, we wanted to grow red seaweed, which is the species that was allowed on the permits. So we selected from the site that, from the Harbor District, the site that we wanted to lease. Right now we are leasing 1 acre, but at the beginning we were leasing just 0.63 acres. And then we deploy our setting, which just consists of a rope that is 100 meters. It's connected to some buoys. And we have 2 long lines, each one is 100 meters. Now, the first species that we decided, and the other thing is we decided to name this farm as HSU Providence. Providence because once you leave the seaweed into the bay, it is going to be based on Mother Nature to nurture this seaweed. There is really nothing you can do, as you will see later on in the presentation. It's going to be Mother Nature who's going to take care of that seaweed. That's why we call it the Providence Sea. Now, the first 2 species that we wanted to grow is Pacific dulse, but also we wanted to grow Gracilaria. And we went collect some Gracilaria from Humboldt Bay, and we were ready to deploy the Gracilaria. But the California Department of Fish and Wildlife asked us if we knew what species of Gracilaria we are going to grow in our long lines. And the reason is because there is a species that is an invasive Gracilaria that is located throughout California. And we were like, well, we don't know what species. We just collected here from Humboldt Bay and we just want to grow it in Humboldt Bay. But they say, well, you have to find out what is the species. And the only way that you can find out the native species from the invasive species is to do some molecular analysis. They look alike, and the only way, again, is just doing some DNA analysis. So we didn't have time to really find out what species we have. And that's why the first project that we decided to grow was the Pacific dulse, previously known as Palmaria. Now the genus has been changed as De La Viera mollis. And our approach was to grow using droppers, which is gonna hang out from the main rope. And why do we want to do that? Well, because we wanted to see what was the depth that they grow the best, the deeper or more to the surface. So that's why we decided to investigate the different depths of growth for Pacific dulse. So just a picture from my graduate student, Erica, deploying the droppers. And what we found out is that the seaweed, if it's close to the surface, is where we have the most growth. And this is going to be because there is more light the seaweed needs light, so that's why we got more growth. What also we noticed is a lot of the seaweed was taken away by the high current that comes throughout the bay. So sometimes the bundle of seaweed could just drift away because of the high current. So that was a disadvantage too that we noticed. But again, we just wanted to see the depth. We wanted to see if Pacific dose was a species that we can grow. And also we wanted to see how safe was for human consumption. And yes, all the values were below, any kind of concern. The arsenic, most of the arsenic, the values seems high, but this is most the non-toxic form of arsenic. Now, something happened while we were doing this first project with Pacific dulls. And it was that suddenly there was some recruits growing in our lawn lines. So they just, Bull kelp was growing in our lawn lines, and we were just surprised to see. And especially because if you are familiar with Bull kelp, and of course you are because you are in Noyo, and you have been witnesses how the Bull kelp has been disappearing in all these places. And yeah, I mean, you are there. We don't have such a large problem as the one that you have there. So that's because we got the recruits, we decided to do a second proposal. And in this time, we wanted to grow Bull kelp in our farm site. In order to do that, we have to get an amendment to allow the growth of Bull kelp in the permits that was approved in 2019. And whenever we are talking about Bull kelp, we are talking about giant seaweeds, something that in the wild, they can grow like 10 centimeters a day. They could reach, more than 36 meters. The way that we compare is comparing about the big sequoias, just providing that ecosystem service, creating the oxygen, removing the nutrients. So all these different services. So that's the next project that we did was again to, first of all, to develop a hatchery specifically designed for Bull kelp. Following was just to start looking into the different string where the seeding, the spores are gonna settle and it's gonna start to grow. So we wanted to compare different of twine or strings. We wanted to compare a more intensive production, having a multi-array system where the lines are close together so we can increase the yield on production versus just the individual systems. And we wanted to see the production of Bull kelp throughout the year. So the first thing was, okay, where do we find information about farming Bull kelp? And the first thing that you need to, whenever you do your research, is like, well, no Bull kelp, but there is a really great seaweed culture handbook by Charlie Yarish from New England that has some of the technology there. But also, Karen Gray was able to find out a publication, the Bull kelp cultivation handbook. On top of that, we also got some training. We took some workshops from Green Wave. But again, all these methods that Green Wave does is more designed for sugar kelp or other kelp species, not really for Bull kelp. So the first thing that we say is like, okay, let's just set up a hatchery just to get familiar for the bull kelp. The first hatchery that we decided to install was a hatchery where the light was on top. You can see those, the spool is just a piece of PVC that is wrapped with twine where the bull kelp is gonna grow. So the other thing is, okay, now what about the light? How much light does the bull kelp is gonna need? And the first thing that we saw is like, well, let's follow whatever green wave is doing, and kind of depending on how fast it's growing, we can change that intensity of light as they grow larger. You know, as you can see here, I mean, it kind of tells you that you need to decrease the numbers of layers that you have or screens to expose the sporophytes to more light as they are growing. And you can see here in this picture, one of those layers that we have in order to avoid too much light to go into the sporophytes. So that's the first thing is, okay, let's design the hatchery. The other thing is, let's get all the permits. What permits do you need to start doing this? Well, the first is to get an aquaculture permit. You know, that's gonna cost about $1,000. The other one is to get a kelp harvesting license. And another permit is to apply for a wild broodstock. A wild broodstock is because you are gonna be collecting seaweed from the wild, and you are gonna be culturing in an aquaculture setting. So you are using that as a broodstock for your aquaculture or your farm. So you need to, first of all, to start getting all your permits in place. And I'm just gonna talk in general about the procedures that we use to inoculate or to seed the spools. So the first thing is we got the permits that allowed us to collect sorite, or the reproductive tissue, from Trinidad, which is the place where we got most of this sorite. So the students go there. Sometimes I go, and then you look for that black patch. That's the reproductive tissue, the sorite. We collect them. We bring it to the marine lab. The next step is to clean, so to separate from the blade, and to disinfect using iodine. Now, if you read the New England or manual, or also Greenway's manual, they're gonna tell you to have to put the sorite between two wet paper towels and incubate them during the night in refrigeration, you know, about 4 degrees Celsius. In that way, it's gonna kind of desiccate, and then you transfer it to seawater after 24 hours, and it's gonna release the spores. What we found out is a lot of the times after the incubation process, the sorite was already releasing the spores, or sometimes they never release the spores. And the thing is, bull kelp, we noticed that the sorite sometimes is just ready to release those spores. They don't need any trigger. And that could be the case. In a lot of the cases, we just bring it from the ocean to the lab, and just add them in a container, and it's gonna release the spores. Another trick that we find out is if they are not releasing the spores, sometimes we just put the solution in the freezer and allow them for about 10, 20 minutes for the water to start becoming frozen, not totally frozen, I mean, just a little, you can see some ice crystals. So that's gonna trigger some stress to the sorite tissue, and it's gonna be like, oh, I'm dying, I'm just gonna release all the spores. So that's also does a trick and release the spores. And another trick that my graduate student Kalani, which is connected here, find out last night, I think it was about 2 a.m. This morning that she was releasing these spores. She decided to use these to have the sorite in this magnetic steering, and as they have more flow, the sorite just start releasing more and more spores. So that's something new that she just discovered, just like this morning. So let me play this video so you can see some of the spores once it's released. This video was taken again by Kalani last night. And I don't know if you can see all those little dots moving around. That's the spores that are moving. They are motile, but they are motile not for too long. So you have to act really quick. Otherwise, that motility is going to decrease. And if you don't seed the spools fast enough, then you are going to be less likely to get a good seeding. The next step is, once you have the spores and you know that they are alive, you need to count them. And we use a hemacytometer, and counting is important. We found out that if you have too much spores seeding your twine, it's gonna be too dense later on to manage in your farm. So that's Marcia counting. You see the counter in her hand, getting an estimation of how many motile spores are. Next step is to dilute the spore solution. At the beginning, we were thinking, oh, let's do like 2,000 spores per ml or 1,500 spores per ml. And as we have been doing this project, we find out that it's way too much. Last night, Kalani decided to seed the spools about 500 spores per ml. And you can see there, Kalani pouring the spore solution into this PVC container that is gonna have the spool inside. This is gonna let it sit for 24 hours to allow the spores to be attached into the substrate. And after that, the spools are gonna be placed into the hatchery. Now, you add nutrients to the water, you do a lot of water exchanges, and just kind of babysit these spores, so in that way they can develop into the gametophyte phase, and later on fertilize and develop the sporophyte phase of those little tiny blades that you can see here. Once you have the twine with a good amount of sporophytes, then you can release them in your farm. Now, let me talk about the farm. In this project, we decided to use a multi-array system that consists of 5 lines that are connected throughout an spreaded bar. As you can see here, there is this spreaded bar and there is 5 lines connected. So this allowed to increase your yield because you are using more efficiently the space. And remember, you are paying about $550 per acre. So you want to make sure that you're using that space efficiently. Just some pictures about the installation of this multi-array system or multi-line system. You can see here the lines going in the water as they are deploying the spreaded bar. Now, at the same time, TNC and Green Wave decided to start leasing and develop a joint farm, which is next to ours. So also here you can see some buoys. The red ones are Green Waves. The white ones are the ones that Cal Poly Humboldt has. One of the differences on our multi-line system is we decided to install this multi-line in a tilted approach, where the deepest part is going to be, depending on the tide action, but it's going to be about 5 meters, and it's going to go all the way to the surface, about 1.5 meters. Why did we decide to do this? Similar to the Pacific Dose project, we wanted to see what is that sweet spot where we have the largest or the most growth of bull kelp. That's why we wanted to compare that. Now, one thing that happened is it takes too long in our previous hatchery to really get that spruce to grow the bull kelp. And one of the reasons that my graduate student Kalani find out is like, well, that's because our light is on the top. What about if we just design a new hatchery and have the lights on the sides, in the 2 sides of the tank? So she just decided by herself, like, hey, I'm just gonna start a new hatchery at the Marine Lab in a place that is gonna be more isolated, that is close to the public. And she plumbed this hatchery. And now this is the hatchery dash nursery system that we have at Cal Poly Humboldt. If you remember, one of the objectives was to compare different twine, different string. And Kalani decided like, well, I'm gonna be comparing the standard nylon twine that we use here at England Marina. I'm gonna compare what the Japanese use, the Kuremona Japanese twine. I wanted to compare a new twine that Gore-Tex is doing. And I wanted to compare something that it could be biodegradable. And Kalani find out like, oh, I'm gonna test hemp twine as a biodegradable option. So again, she collected the sorites. She was able to grow the sporophytes. As you can see, the different percentage on growth depending on the different twines. And what she found out after all the work that she did is that this new hatchery, it just cut almost in half the time to grow these sporophytes than our previous attempt. So overall, you know, she found out that nylon was one of the best twines to use. And that's very good to know because this is the cheapest, one of the cheapest options that you can just find locally. You can see some of the sporophytes growing on the nylon at 5 weeks. And then you can see them larger at 7 weeks. Another thing that we found out is hemp twine. Hemp sounds good, it's sustainable, it's biodegradable, but what happened is it biodegrades too fast. So what happened is once we release them or we try to release them into the bay, it was just breaking into parts. So it's good, it's biodegradable, but for our purpose, it's biodegradable too fast that it's not really useful for a kelp farm. So once you have your spools ready, remember the next step is to deploy them into your system. So you see here, Kalani deploying the systems. for her project on the 4 types of twine. Now, next, I want to talk about some of our preliminary results from the several attempts that we have been doing. So in our first attempt, that was last year, we were able to grow the bull kelp in our system. In fact, we have 2 seasons, and you see here Clanny holding that bull kelp, little tiny bull kelp. And again, I remember that I mentioned the importance of densities during the hatchery phase. Well, what we find out is if you have too much spores seeding your twine, you ended up with a lot of bull kelp that is gonna compete themselves, and you don't get this huge bull kelp such as in the wild, like you have one after another. So this is the second trial. We outplanted the seedlings on February the 26th. The kelp grown in the deeper part, the larger, compared to the shallower part. So contrary to the Pacific dose that we found by using the droppers, that Pacific dose like to grow at the surface where they get most of the sunlight, bull kelp, on contrary, they like to grow deep. That's where we got the largest individuals or the largest growth. And of course, it makes sense, you know, bull kelp is in deep water. You don't really see too much bull kelp in shallow areas. Again, some pictures. So the students, Kalani and her group goes every week, measure the bull kelp. So this is our results from last year, where we outplanted the seedlings in February and we harvest in June. It was 116 days and we were producing not too much, less than 2 pounds per foot. The reason of this really low production was because we waited for too long. We waited once June, late May started, those temperatures in the bay sometimes were above 16 degrees Celsius, and suddenly the kelp start to deteriorate. Some of the problems also that we encountered during this first project was as the bull kelp grow, sometimes it get entangled with the other lines and it just make a mess, like it just entangled. Here is a picture on the left side on the multi-line system. As you can see, they entangle. And here on the right side, you can see how warmer temperatures are going to start to deteriorate the bull kelp. So right there, we found out that definitely bull kelp is not going to be a crop that you want to grow in the summer in Humboldt Bay. But, during the project, we have been able to grow larger individuals, such as this one of our largest individuals, about 7.6 feet. Overall, we were able to produce close to 500 pounds of biomass last year. Now, let me talk about our most recent approach or project, which has to do with the different twines. And this just happened about 3 weeks ago. We deployed or outplanted the seeded twine, the different twines. Again, the nylon, the Japanese twine, the kuremonan, the hemp twine we couldn't deploy because it was already breaking apart, so it was not seeded, and also the Gore-Tex twine. But what happened is we just outplanted after the biggest storm that happened, at the beginning of this year. In fact, Green Wave side, they seeded their lines before this big storm happened. And unfortunately, all the fresh water that ended up running into the Humboldt Bay make the bay really brackish. About 20 ppt on salinity. So if you know the ocean is about 33, 35 ppt on salinity. So now we are talking about brackish condition, and definitely Bull Kelp did not like this brackish condition. And that was the case of our first attempt. We, after all the work that Kalani did on collecting the sorais, taking care of them in the hatchery, nursery, and then outplanted, and suddenly having Mother Nature again, these brackish conditions, there was nothing we can do. The bull kelp, the sporophytes, it seems that they did not survive the brackish condition. But one of the long line systems that we outplanted 2 weeks later, it has some growth. So you can see the little sporophytes, the little blades growing along the twine that is wrapped around the rope. So we are expecting that in our long line, we are expecting some growth, but unfortunately in the multi-line system, it's not the case. And that's why Kalani last night had to start over her experiment on comparing the different twines. The good thing is this time she's comparing also a Korean twine. Korea is the country that started seaweed farming about 1500s. Now, going back about what we can do with all this that we have been doing, and also to introduce another project that we are doing. So in order to recover some of these lost kelp forests, there is attempts, to remove as many sea urchins as possible for the ewing. But also there is another attempt that is going to be outplanting some of the kelp. One of the techniques known as the green gravel approach that was developed in Australia with other kelp species, using gravel and grow the kelp on it. So we decided to jump into the boss of the green gravel approach. And I'm talking about the boss because we are not the only institution doing green gravel. There is Sonoma State, and there is other institutions doing the same approach. So the first, I think, this is the project for my graduate student, Marcia, was to find out what type of substrate is going to be better to use. Do we want to use, like a pebble, a rock pebble, or do we want to use some kind of a ceramic tile that is going to be easy to drill or just to sit tight in the bottom? So that was the initial idea of her thesis project. You see there, Marcia inoculating the tanks with the spore solution. Her project was to compare at the beginning, like 3 different temperatures. How well is going to grow at 10, 15, and 20. But also, of course, to compare the 2 different substrates, how well it's going to grow in a pebble, in a rock, versus the ceramic. Overall, the ceramic was better. You can see better on a ceramic. But during that experiment that she conducted, she realized that 15 degrees Celsius, it just speed up the process. The bull kelp was growing really fast. 10 degrees, it takes a little bit longer, but then on the 15 degrees Celsius, they grow, and suddenly they start bleaching, or the blades start to fall apart. So they need probably a larger system or more flow. But then what about that 20 degrees Celsius? It seems that that's really warm for bull kelp. And what happened is, it was suddenly a lot of contamination from from the green algae. As you can see, the colors of the rocks, it just look green because, yeah, there was contamination from green algae. But while she was observing on the microscope slides that she has in each of the tanks, she noticed something that looks like a gametophyte of bull kelp at 20 degrees Celsius. And what happened is, previous publications say that after 18 degrees Celsius, there is no growth of gametophyte.
I don't think he's Indian.
Yeah, I think he sounds Spanish.
So,
I don't know if they can hear. Oh, I want to be muted.
okay, so that was amazing to kind of find out that bull kelp could develop all the way to the gametophyte phase at 20 degrees Celsius. Something again that previous publications did not find out, or for that no more than 18. So this kind of changed the way that we look at this project, and we decided, like, forget the green gravel. Let's just focus on temperatures, and let's just see how well it's gonna grow. And let's try to shoot for 10 degrees, 18 degrees, and 19 degrees. Well, in fact, we wanted to do 20 degrees, but the heat pump was reading 20 degrees, but internally in the tank, it was 19 degrees. So the first one was, okay, let's do a humble trial, just get some sorite from Trinidad and expose them, see them in the microscope slides where we can really easy to see or observe the development of the bull kelp. So 10 degrees, perfect. They develop into the sporophytes. Then on 18 degrees, we start also noticing some sporophytes growing. And again, this is surprising because previous publications, they did not notice that 18 degrees was viable for bull kelp. And then on 19 degrees, there was not really sporophytes, but there was a presence of the gametophytes. So this means that the spore was able to keep developing into a gametophyte state, which was also new, something that is new. The other thing was, why don't we try to see and compare bull kelp from different locations within California. So we were able to get some sorite tissue from Monterey. Unfortunately, just one sorite tissue is the one that released the spores. And we were just repeating the same experiment. This time, one of the difference was the Monterey trial, the bull kelp from Monterey, it just grew really fast compared with the one in Humboldt. Also, another difference is that the 18 degrees Celsius, we didn't see that or mature sporophyte. It was more of a young with not that many cells division as a multicellular one that we have in Humboldt, but still, you know, we saw some small ones. And then on 19, we still see the gametophytes, but not any sporophyte. So as you can see, there is already some variation And it's interesting to see that Humboldt was able to have a more developed sporophyte at 18 than Monterey. And we want to repeat this. We are trying to see if you and work with Sonoma State and see if we can get some sori from your area. And also we wanted to see if we can get some sori from Baja, just to see the difference on the developmental stages at the different temperatures. So that's just one slide showing you the growth of these sporophytes. We have been working in other projects involving undergraduate students. Another project that we have that we were doing was to compare LED lights against fluorescent light. Unfortunately, this project, it just become too warm, the system, that all the schools died, nothing really developed. But you know, like we are trying to do research. Something that also we want to do probably in the future is to do salinity tolerance and see how well does they grow at brackish conditions. So overall, the benefits of this project, I mean, has been an excellent opportunities to train the students. We deployed the first commercial scale seaweed farm in open waters in California. We are expecting that this will create a new industry, generate jobs. It could help an additional crop for some of the shellfish farmers and to develop new products and markets. Overall, the representation of Cal Poly Humboldt in aquaculture conference has been increasing over the years. And also we have been working with the California Seaweed Festival, and I strongly recommend you to join and come. We have a festival every year. Last year was in San Francisco in Tiburon. This year is going to be held in San Diego, and we are also planning to have a field trip to Baja to see some of the seaweed that they grow over there. The next project that we have in line is to change the species. We want to grow Alaria. Alaria is a species that everyone likes. It has a better taste. And that's going to be the next species that we're going to be growing by the end of this year. So I had the opportunity to be in Alaska this summer, and I witnessed, you know, how California could become in the future. We have the ocean, we have the space, we have the technologies, and there is no reason why not to start growing more seaweed throughout California. This is, again, it's an extractive species. It's providing a service to the ocean. We also, last semester, we have Charlie Yarish as our invited speaker. He's kind of the father of the seaweed industry in the United States, and we appreciate his visit. I just want to say thank you for all the funding that we have been getting from the CSU Agricultural Research Institute. We get also, that grant requires match, and the matching is coming from the USDA HSI H. Alegranos grant, also the Western Regional Aquaculture Center. I want to acknowledge the sponsor program foundations and the Humboldt. I mean, the list is there. So definitely, this is not, I'm not the one doing all the work. My students are the ones doing the work. So I'm just here talking. So that's it. Thank you.
Bravo, that was amazing. Gosh, thank you for sharing so much with us. It's just inspiring and exciting. Informative talk. Might kelp aquaculture learnings lead to solutions for urchin barrens in the wilds? Kimberly asks. So, yeah, do you want to talk about that a little bit?
Yeah, definitely. One of the things that we think that it could help is outplanting some of these row where we have all this bull kelp growing. So it's just a matter of logistic. We can use Humboldt Bay as a nursery to start growing some of these bull kelp juveniles and find a way to outplant them in Mendocino or Sonoma. Yeah, that again, everything has to do with permits. So it's applying to other permits, see if this is something viable. But we do this in oysters. Humboldt Bay is a nursery area for oysters, and we grow these oysters, like even in Washington, but it's nursery here.
Yeah, the permit process sounds daunting, but I'm sure that, it would be worth it. And looking at the topography, I mean, you've got Humboldt Bay, and here in Fort Bragg, if we try to tackle some of those thoughts, thinking about where and the different, we don't have a big bay. We've got the Noyo Harbor, which is pretty small. And then other bays, but in terms of estuaries or bays, probably not, but lots of bays without a doubt. And then lots of them have got, already lots of urchins in them. So definitely not a new conversation, but jumping over those hoops, through the hoops. Here's a question, wondering about how concern for disease in seaweed aquaculture compares to the Finnish aquaculture. Have you noticed disease in the farmed seaweed? So comparing it to the wild.
We have not noticed any disease, and I'm not aware of a disease on bull kelp in a specific. I mean, we saw that higher temperatures is going to start to deteriorate, and yeah, the nematocysts start to just lose that beautiful form, but it's just the higher temperature. It's not really because of a disease.
Mm-hmm. Right. And then what were you cleaning off with the iodine?
Yeah, so because we're getting the sorite out of the wild, you get all these different type of parasites or zooplankton, I mean, microorganisms. So you want to try to sterilize the sorite, so in that way you don't pass that contamination to the hatchery. So you are trying just to remove some of these pathogens, potential pathogens, yeah.
Aha, clever. And it doesn't, the spores, the sorry don't mind the iodine. That's interesting.
Yeah, well, we don't let this sit for too long, normally 30 seconds or 1 minute. So it's not like they stay there for a long time. It's just a little dip.
Right. What about the paper towels that you were saying? Are they're damp paper towels, but they're damp with seawater, with the water that you're using. Right. You're not.
Well, we use sterilized seawater to wet the paper towel and also to inoculate or to dilute the spore solution. We use distilled seawater.
That makes sense. Someone's wondering if you've experimented with spore bags.
Spore bags, like the ones that Moss Landing is doing, where I think that's the question, where they just have the reproductive tissue, the sorites, in a bag, and then just release them or outplant it. So in that way, it's just going to start releasing the spores. We have not been doing that. Again, our approach is more farming. That was the initial approach. Rather, I mean, nowadays we are doing, more of a ecology or restoration. But yeah, we have not been doing the spore bags.
Right. Right. Let's see. Did you have any sensor loggers in your nursery temperature, salinity, pH, or other?
Yeah, so the students, they measure water quality every week. So they measure nitrites, NO3, they measure phosphates. They are adding nutrients into the system, F2 nutrients. But probably we want to change for probasole next year. But yeah, they're adding nutrients, they are checking water quality, and making sure that they have enough nutrients to grow.
Sure. Let's see. Great questions, everybody. Someone asked me to address Rhoda's question, and Rhoda said, did any kelp reseed below your nursery area or nearby? Like fall from the area and start growing on substrate nearby?
Oh, well, the thing is, the visibility that Humboldt Bay has is very limited. So we don't really know, if some of the seaweed that we are growing ended up in the bay, and if it's growing, we have not, we have not seen nothing like that yet. But again, yeah, the visibility is really bad.
Right. Yeah, so one thought here, like if we were to put the spores in the bay where all the urchins are, immediately it's attractive to see the little holdfasts on something that is above the rocky intertidal or the rocky bottom where the urchins are. So like having them suspended up high looks very attractive too. Like that. And I love seeing the little kelps grabbing onto, sometimes when you're walking on the beach, you find a beach cast chunk of something or a different seaweed, and you can see all the intermingling little holdfasts.
Mm-hmm.
What did you do with the 500 pounds of kelp that you grew?
Yeah, so that's something of a limitation, because if we want to develop a seaweed industry in California, we need to start thinking big, like what they are doing in Alaska or whatever they are doing in the East. For example, Alaska, there is Barnacles, a company that develops salsas, and they do like prickles. There is even some popcorns that they enhance with seaweed. In our case, because we already work too much on the farming side, we don't really have time to explore the processing. Pretty much we just donate it to a local farmer that he do amendments on soil. So he just combined the Bull Kelp and he does his soil amendments. But yeah, unfortunately, we want to see this, like on the East, they even make beer out of seaweed. So it's like, well, why don't we do something like that around here too? But yeah, for us right now, we just donated to a local farmer.
You could see that Humboldt State could probably make a pretty mean kelp beer.
Yeah.
And it would be supported. If you turn something into beer, it's probably going to go. Hey, Barb, do you want to, I think you can unmute yourself there and ask your question.
Sarah, thanks, Rafael. That was just an amazing presentation. I'm just so impressed by the sense of wonder and awe that you and your students have, and just trying to explore the various, layers of discovery when it comes to this. And it sort of feels like the seaweed is the Wild West of the science right now. So it's just wonderful to hear and see your work and the protocols also, which sort of really inspires me as one of the beach surveyors to get dirty and get out there even in the windiest and coldest days and do our work, because we can see that you're really digging in. My question for you is just about companion planting. Like on land, we think about how we can plant other plants next to each other that are beneficial to one another. So marigolds next to your beans, and the various insects and stuff that work on and against each other are working together in one spot. Have you thought about companion planting or seen any effects of companion planting when it comes to seaweeds? So planting one type of seaweed next to another, or oysters amongst another seaweed, or anything like that?
Yeah, excellent question, Barb. Yeah, this is the area of what is called integrated multi-trophic aquaculture systems, also known as IMTA systems, where you have multiple species, and the byproduct for one species becomes the feed for the other one. We have been talking about having oysters or scallops growing on the same lawn line. In that way, they could fertilize our seaweed. The seaweed is going to uptake those nutrients, it's going to absorb, and the seaweed then is going to remove that CO2 that is not beneficial for the development of the shell. So there is a benefit for the bivalves to have seaweed, is removing that CO2, but also there is beneficial for the seaweed because it's going to uptake the nutrients. So it's on our radar, but the thing is, again, permits. If you want to add a different species, now you have to do an amendment or a new permit again, where you need to lay down what is going to be your setup, your farm. So again, it's yes, we want to do that, but we have not done it yet. What we have been done in inland is at the Marine Lab, we have sablefish, and the nutrients that is coming out of the sablefish tanks goes into a seaweed tank where we have Pacific dose, and we have that same scenario that you're talking. We have it on land where we have a recirculation system growing sablefish alongside Pacific dose, and also Olba. We have Olba too.
Will you be doing any cloning? And do you think the different locations you took kelp samples or different strains of DNA wise? Sorry, I botched that, but different DNA for the different kelps and bull kelp in different locations.
Yeah, we have not been doing any kind of DNA analysis. Again, this project, Marcia's project, is kind of a piggyback from our big grant. It's not like we have a lot of money for that project in a specific. One of the things that happened is one of the committee members is like, how can you assure that the gametophytes that you see at 19 degrees Celsius is from bull kelp and not from a contamination?
Right.
And because of that, we have to do DNA analysis on the 19 degrees Celsius just to make sure that what we see is coming from bull kelp. We know it's coming from bull kelp, but again, we just want to double check with DNA analysis.
Sure. And then one last question, and then I'll let you go. But is your hatchery an open system?
Open system, like a flow-through system, not. And Kalani is here also. I don't know if we can invite her. She's the hatchery manager, my graduate student. I don't know if she's listening or not, but it would be nice. Hey, Kalani, I don't know if you want to talk about the hatchery and what you have been doing.
Yeah, so the hatchery that we have set up in the Telenoca Marine Lab, we have it in what used to be a small ornamental room. And so we plumbed the system for it to be a flow-through recirculating system, but it is technically a closed system, meaning that we have our holding tank, which we're just using a cooler and putting our sterilized seawater and then having it run through 8 tanks that are all connected. So it would technically be a closed open flow-through system.
Awesome. Thank you for answering that. And yeah, all things aside, just the focus and the goals that you set and the success that you had with what you set out to do is phenomenal. You know, really beautiful to see the kelp, the baby kelps. And my favorite picture, I think, is you, Kailani, holding the little teeny tiny lollipops as they started to grow. It's like just beautiful, and really, I know that's not easy. So congratulations to you all for doing this, and much success getting through permits and getting funding in the future. It's great work, and we all are so appreciative to you for coming and giving us this time. And here's just another great example of Noyo bringing some great stuff to you all. So continue to support our organization. So I guess that's a wrap. Unless you had anything else to say, Rafael.
Yeah, if you want to visit our hatchery, we are in Trinidad. Kalani, you know, like I mentioned, she seeded these new spools. Probably they're gonna be here for about a month before we are planted. So unfortunately, we cannot offer a visit to the farm site because of the liabilities on the boat, the university boat. But you are always welcome to visit the hatchery and see the spools growing.
All right, thank you so much, and everyone have a great night.
Bye now.
Thank you.